Hi everyone, we're back again with another video tutorial, this one on the use of selective and differential media. You've learned a bit of the theory of this, and we'll talk more as we go through the lab about the theory of this type of media and how it works. And there's a fun added bonus. So we're not only working today with known bacteria, we are going to do some of that, but we are also going to work with some food samples. Here they are down here. So these ones are going to be a very expired yogurt sample that's expired a year ago. Don't ask us why it's still kicking around. We keep it entirely for microbiological purposes. We also have in that jar, the reddish liquid in there is juices from meat. I believe, was it sort of chicken or a mix, Sabrina?
Chicken, vegetable, and fine bone raw dog food.
Excellent. So this is being left down at room temperature to increase the chance that we'll get microbial growth, and we're pretty sure we do have that. And last but not least, we have, we'll call it parsley juice. So this happens when you leave parsley in the fridge for too long, which we also do purely just to create cultures for you guys. Never accidentally.
No.
And you've probably maybe seen this yourselves. It starts to kind of liquefy. This has had a little water added to it, and then it's left down at room temperature to enhance any kind of microbial growth as well. So we're going to be plating these, spread plating these onto some selective and differential media, that colorful media that you've seen in kind of lecture slides that is there. We're going to look at some supportive media at the end there. And actually, you know what, before I'll talk through those as Sabrina gets to work on doing some known bacteria on those selective and differential media plates. So one of the things that you always want to do when you're looking at a food sample where you don't know what type of bacteria are in there or what you might expect to see, you want to do some known bacteria. So you know that you're seeing the expected results on those selective and differential media plates. So we are going to look at 3 types of known bacteria. And we've had Melinda dilute these out a little bit for us so they don't look as turbid as you typically would. This way we can get nice kind of colonies forming on the plates. So the 3 bacteria we're working with here are, and I can zoom in a little bit to see, although it's still going to be tricky to actually see them, but there's our tubes. So we have Staphylococcus epidermidis, sort of the closest to the Bunsen burner there. We have Staph aureus, or Staphylococcus aureus, and then we have E. coli, or Escherichia coli. So you guys, I'm not going to say what type of bacteria these are as we go through, and that will be a little exercise for you guys to do to see sort of what you would predict to see on these selective and differential media plates once we actually culture these bacteria on them. So you can see that Sabrina's just getting her loop there. She's flamed it, got it nice and red hot. She's letting it cool right in the sterile zone. And she's going to grab out a plate, and I'm just going to, actually, yeah, you can see that really clearly. So in this case, we're not doing a full streak plate. We want to see growth of these bacteria, but we're not so interested in getting isolated colonies. We know we have a pure culture here. Isolated colonies are great, but we're not trying to get a whole streak plate of them. So she's just going to actually, and you'll see her do it, just do a squiggle inoculation of these plates where she's simply going to draw a little squiggly line back and forth to inoculate that plate, and she's divided it up into 3 sections so she can put all 3 of our known bacteria on that plate. So you heard the vortex go, so she mixed the Staph epidermidis. She's going to now grab a loopful. She's doing nicely her aseptic technique there with making sure the tube is flamed before and after. She's now going to inoculate that. So pick it up by its base. And I'll just try to zoom in there for you guys to see that. And there she goes. She's just going to go back and forth in that 1/3 of the plate that we've sectioned off until it's done in a nice big squiggle, and then flame the loop again as she's doing right now. So this is a really great way when you're not really, you don't need 10 or more isolated colonies, to not use too much media, right? Because we're going to literally inoculate on that plate. We're going to use 3 bacteria on that plate instead of just having one type of bacteria on that plate. So she's going to work through all the media there. So the first plate she did was actually something called PEA, and this media is selective for actually I'm going to stop myself because I'm not going to say. She's now going to inoculate staph epi, that's what she just did, now onto an MSA plate, or a mannitol salt agar plate. So that was the nice cherry red plate you just saw. She's now going to grab another plate. This is a deep purple color, and this is the eosin methylene blue, or EMB agar. And she's doing the same type of bacteria. So she's going to work through all of the different media types we're working with today. And she's going to inoculate the same bacteria onto each of them. Then she's going to go back and start over and work through those plates again, doing that same squiggle kind of inoculation of 1/3 of the plate. So there, she's doing her third for the EMB plate. And those are our 3 actual selective and differential medias that we're using today. And like I said, I'm going to keep it secret what they do. You can look it up and figure it out, and figure out what your expected results would be of these known bacteria on these plates. The last media type you do, and you would always do this in an experiment, is a supportive media type. So this is not selective and differential. It's not going to prevent the growth of any certain bacterial types. You should see a wide variety of bacteria are able to grow on it, and these 3 should all grow on it. So we should see our Staph aureus, our Staph epidermidis, and our E. coli all growing nicely on our tryptic soy agar plate that she just inoculated with the Staph epidermidis. You want to do this because when you have selective media, you're often preventing the growth of bacteria because of what's in that media. If you don't confirm that you have bacteria that are growing well on a supportive media, it's hard to make conclusions from that selective media. So you always include a control, essentially, where you have some supportive media that should let your bacteria that you're analyzing grow. So now Sabrina's working her way back through these plates with the next bacteria type, or you just guessed? She's on to Staph aureus now, and she's going to work her way through those through those different media types. Obviously, when you're working with multiple known bacteria, or multiple different samples for that matter, your aseptic technique becomes very important in order to prevent sort of cross-contamination. So you want to make sure your loop is fully sterilized before you dip into a different culture of bacteria. Now she's working with that lovely cherry red mannitol salt agar plate and inoculating with Staph aureus. Through that. This takes a little bit of time, like a lot of things in microbiology. There's a certain element to the lab work that's a bit repetitive, and this is certainly one of them, but we just have to work our way through all of these in order to get our known bacteria onto the plates. There's the EMB being inoculated, loop being sterilized. Same pattern, always mix before so that we know that, that we have those bacteria nicely resuspended in that culture. One of the things that can happen is, they are essentially cells floating around in a liquid. They will eventually settle to the bottom of that tube. And if they've all settled to the bottom of that tube, you can get the impression that there's very few bacteria in that tube, that test tube, unless you actually do that good vortex before you get started. So you can start, if you want, thinking about what you would expect to see on these plates after this inoculation and figuring out, because you know from lecture, you might already know which ones of these are gram-positive, which ones are gram-negative, but a lot of the selective media is selecting for one or the other. So selecting for a broad group of bacteria, such as all the gram-positives or all the gram-negatives. Whereas when you have the differential media, then you're looking usually more at subtle differences, more subtle differences between more closely related groups of bacteria, where they will both grow on the plate, right? That's the whole differential part, is that you see both colonies of bacteria, or both types of bacteria grow, but their appearance is going to be different depending on what species they are. Working our way through now with our last known bacteria. This is the E. coli. Going on to the nice mannitol salt agar plate. See how Sabrina's had that loop right ready to go. She's going to flame. Just remember, as long as your loop's sitting in that sterile zone, you can sort of sterilize it and let it sit there for, you probably wouldn't want to let it sit there for 5 minutes or more. You'd want to re-sterilize it then, but if it's just for a minute, just let it sit there and it's going to stay sterile. And last but not least, we're going to do our E. coli sample on our tryptic soy agar, so our supportive media that should grow pretty much many types of bacteria. Definitely E. coli, as you've seen from previous labs. Tryptic soy agar is a bit of a workhorse of the microbiology lab in that it's used for growing many types of bacteria. And there, so now we have our, what I've called squiggle inoculation, so our squiggles on each of those plates of our 3 different bacteria on our 4 different types of media that we're working with today. All right, now we get to go on to the good stuff. So here we have plant juice, parsley juice. And one of the things that we would find if we simply just threw this onto a spread plate and spread it around is that we would end up with a lawn of bacteria. We would have bacteria everywhere, most likely. But if you dilute it a bit, that's going to let us see colonies or just reduce that lawn of bacteria to a point where we can get a better idea of what's going on that selective and differential media plates. So Sabrina's going to start with tenfold dilution, also called, as we discussed in lecture or in the virtual lab series, a 10 to the -1 dilution or a tenfold dilution. She is working with the micropipetter that you've just learned about, and what you saw her do there, and maybe I'll have you do that again one more time, and I'll just zoom in. See how that black switch is moving from one side to another, from the closed form to the open form? That's going to let her adjust the measurement there. So she needs to move it so that it looks, or so that she's pulling up, in this case, 1 ml of our plant juice. And at first, you're not going to be able to turn that dial, the blue dial, and that's where that lock comes in. So you need to unlock these micropipetters first, adjust the blue dial until you see 1000 in the window there, which indicates 1 mil on these micropipetters. And then you can get your tip on, right? So she's getting their tip on, making sure that box stays closed until she's right ready to open it, closing it up as soon as she's got that tip out. Always a little awkward with these giant jars. It's a little weird to have our home containers in the lab, but that's when you do these food kind of labs, that's what happens. She's pipetting up 1 mil slowly and steadily, right? Not releasing her finger off that thumb quick or off that button quickly, or thumb off that button quickly so that the air gets into the or into the pipette tip. Now she's going to release it out, making sure she's kind of puts it along the side, blows the last little bit of liquid out, and then that pipette tip goes into the biohazard container over there. Now she's got a lovely diluted solution of plant juice, and there it is getting vortex so that it's really, really well mixed in there. It is always a little funny doing these because there's usually a few chunkies and things that you normally wouldn't see in a typical sample, but there it is. You can see the effect of the dilution. It's much less sort of turbid and dark than it was before, cloudy and dark. All right, now she's readjusting. So we are going to spread plate this, and when we spread plate, we are going to add 100 microliters to the spread plate. So remember, 100 microliters is 0.1 of a milliliter, and so we need to adjust that down so it reads 100 in the window rather than 1000 for 1,000. So now she's set that up. Always gonna lock that up again so it doesn't get bumped or jiggled and readjusted. And she can get ready to pipette now using that micropipetter onto the surface of a spread plate. I think she's grabbed our first one there, which is our PEA media. And she's just going to pull the lid off there of the tube, flame, reach in, pull up 100 microliters. You'll notice, I mean, I'm not going to be able to zoom in to show that exactly, but what you want to keep in mind when you're pipetting is always, does the amount I'm pulling up make sense? So you'll notice when you switch from 1 mil to 100 microliters or 0.1 of a mil, the dramatic difference in what is going up into that pipette tip. So it's just really not very much that you see going up with 100 microliters, and it's going to fill that pipette tip quite a lot when you're doing 1 mil. All right, so Sabrina's getting ready to do the spread plate. So she's just waiting now. She actually did the quick flame off of the ethanol. You can see the ethanol beaker hiding behind the Bunsen burner there. Yeah, there it is. And now she's ready. It's probably cool because remember, you're never getting that spreader really red hot. You don't have to hold it in the flame because it's being sterilized essentially by the 70% ethanol. Now she's ready to spread. So there, we got the plate going around and round. Spreader goes back and forth for the spread plate. And then the very end, you can kind of go around the outside, and it should be good. And we'll have to see what happens when we grow this, incubate this mix of juices from the parsley that's, like I said, purely for lab purposes. I left way too long in the fridge. Okay, next one, we're gonna move on. So this is, once again, we have to work our way through all our media. We're gonna move on to the mannitol salt agar plates and pull up 100 microliters, 0.1 of a mil from the 10-fold diluted plant juices. Put that into the center of the MSA plate. Discard into the biohazard for the pipetter tip. And flame off the ethanol. There it is up there. Always watch it to make sure that flame is out before you start testing it on your plate. test it onto the lid there and watch. Help cool it down. Often if you see a puff of kind of condensation that appears on the lid, that's an indication it needs a bit more cooling. Test it on a spot in your plate where there's no bacteria. Once again, if it sizzles or if it starts to melt the agar, it's much too hot still and you need a bit more time. But it looked good, so Sabrina's going ahead with the spread plate. And she's really nicely getting that across there. You know, you don't have to spread forever to have it work well. So the turntable definitely accelerates that too. Okay, so we're gonna vortex. There's the hand going across. You can hear the vortex going. You can see how Sabrina's really careful, right, to shut that box of pipette tips every time. So if you're pipetting, you know, anything in the microbiology lab with those pipette tips, you want to keep those as sterile as possible. They've all been autoclaved, but you want to open it for the most brief amount of time as possible and shut it up as quickly as possible, and also keep it close to that flame, working in that area that's sort of what we call the sterile zone. Ready to go with the spreader again. Testing it up. And Sabrina's going to now spread on the EMB plate, the one that looks like sort of grape, very purple grape juice. And that looks good. So we've got 3 of our media plates. We've got all of our selective and differential media plates done. And then of course the last one, just like we did with the known bacteria, is our supportive media, our tryptic soy agar. And so we're going to do our last measurement or aliquot of 100 microliters of plant juice, of our 10-fold diluted plant juice, out onto that plate. And spread that in, and then these guys are going to go into an incubator at 35 degrees Celsius for 24 hours in order to see what we get in terms of growth. And then we can, you know, we'll do some filming for you guys and we'll show you what the results look like and what type of bacteria. And then you guys can kind of think about, how do those plates work? What's the selective part? What's the differential part? And so given what we see on those plates, what are the most likely suspects in these food samples? And of course, comparing to what we see on with the known bacteria, because that'll give us some hints too about what we might have in our food samples. One thing I'll mention now, just as Sabrina does the very last plate, is the dilutions. So here we made a very simple dilution. We did a 10-fold dilution, also called the 10 to the minus 1 dilution or 1/10 dilution, where we put 1 ml of our plant juice into 9 ml of saline to give us a total of 10 ml. This, as you go forward, you'll see we're going to make some much more elaborate dilution series than that, where we dilute all the way down to, say, 10 to the minus 10. So this is a simple one. We might do a few extras for these food samples, not on video, but just for you guys, so in case we still have a lot of bacteria with our 10 to the minus 1, because it's not uncommon to have quite a few bacteria in there. So, especially the meat juice. You'd be glad that this is not a full sensory experience, because you do not want to smell that. Anyway, I think going forward, you guys will get experience doing these kind of dilutions. This is just a taste of kind of the first steps of making a dilution series. And
the wrong time to use it to taste. I certainly hope not.
Yes, no, we're not tasting anything in this lab. No tasting is ever involved.
Especially not this.
Especially not the meat juice. Anyway, guys, hope you check back in when we have the results of this, and we'll show you the video of that. All right,
Awesome.
Thanks, guys.
Hi everyone, we're back with our results from the selective and differential media where we plated out some disgusting food samples and looked at how they grew. I have taken a little bit of a look at our results and we have some interesting ones. I want to start off with our positive controls. So this is our positive control on TSA. And something that I'd like to point out is we do not have any growth of staph epi. So Staphylococcus epidermidis, we just straight up don't have any growth of it. So this shows us that we had a little bit of a problem with our culture. So we are going to have to redo our positive controls and get a new set of data for them. In any case though, we can see E. coli over on where my fingers are, and Staph aureus down at the bottom there. And that's on TSA. If we look at MSA, we actually didn't have growth of any of our organisms, and that is not what we would expect. So this definitely shows us that we need to have a better inoculation of our positive controls. On PEA, we only have growth of E. coli, and those are little teeny tiny colonies. Let's see if we can see them there. Yeah, they're just little bitty dots up by my finger. And then on EMB, we also only have growth of E. coli. And those colonies have this really cool metallic green sheen to them. So that's something that's typical of E. coli when it's growing on EMB. Now, here's the fun part. Let's look at our samples, our disgusting food samples. So first off, we had yogurt. And when we just did that single dilution that we showed on camera, we only saw growth of one single colony on TSA. And all of our selective and differential medias, we did not see any growth whatsoever. The only little things there, those are just bubbles that were in the media. So our yogurt, even though it was expired for over a year and a half, it actually didn't have any growth going on in that whey portion of it that we took a sample of. And surprise, surprise, when we did a further dilution, again, no growth on any of those plates. If we look at the parsley juice, we can see that on TSA we had a complete lawn of bacteria. On PEA we had a complete lawn of bacteria. On EMB we had a complete lawn of growth. And then on MSA, this was kind of cool. We have some colonies that are kind of a yellowish color, and they've actually changed the media to a yellow color. So that's an important feature on MSA, so that'll be something to look out for when you're doing your assignments. We also further diluted our parsley juice, and here we can see a little bit more spread out results. So on our TSA plate, we still have basically a complete lawn of growth. However, we can start to see some different colony types, and I'm seeing at least 3 different types. I'm going to give this to you guys as a photo as well, and hopefully you'll be able to see some of that. On our MSA plate, we still have just one of those colonies that has that yellow clearing around the media. On PEA, we can now start to actually see some individual colonies. And again, it looks like we've got at least, I think we've got at least 4 or maybe even 5 colony types on there. So that should give you some interesting information to look for. And then on EMB, we still have basically a lawn of growth. But again, we're starting to see some distinct colonies. And even though these are super tightly packed together, I can see that none of them have that metallic green sheen. And I can also see that we have at least 2 different colony types. One of them is a smaller colony and the other is actually quite large and really bright pink. And now the most disgusting of the samples, we have our meat juice, the juices from raw dog food that had been left out. So on our 10 to the minus 1 dilution, that first dilution there, again we've got basically a complete lawn of growth. On MSA, we have no growth at all. On PEA, we can see that maybe my spreading technique wasn't as great as we would have hoped, but we still have essentially a lawn of growth. And on EMB, first off, I want to point out that we actually had a change in media color up at this one edge, so that should be something interesting to look for. And then here we've got, you know, our bacterial colonies are super closely packed together. It's basically a lawn of growth, but again, I can see that we've got at least 3 different colony morphologies, and some of those are really mucoid. None of them have that metallic green sheen though. So that's why we need to now look at the ones that were diluted a little bit further. On TSA, again, we've got growth throughout the entire plate, but here I can start to see that we've got at least 3 distinct colony morphologies. MSA, we didn't have any in the more concentrated sample, so we don't have any on this sample either. For PEA, again, we've got almost uniform growth all around, but this time we're at least seeing distinct colonies. And we've got at least 2 different colony morphologies on there. And then for EMB, we're a little bit more spaced out. And here I can see it's spaced out enough that I can tell that we actually have at least 4 distinct colony morphologies on those EMB plates. So I'm going to get these photographed in still for you, and then you'll have a great set of data to look at and do your report on.
Hi guys, I'm back to show you the results of the known bacteria, E. coli, Staphylococcus epidermidis, and Staphylococcus aureus on our selective and differential media. We'll start off just looking at our supportive media. That's our tryptic soy agar, as shown here in the screen. So you can see that we have all 3 of our known bacteria growing nicely. We have our E. coli there, our Staph epidermidis, and down here at the bottom, our Staph aureus. You can see their colony morphology is a little bit different. Staph epidermidis has very small round colonies. Staph aureus has a much more golden color than the others. And E. coli has the larger, probably the largest colonies, very creamy colored. So that's very typical for what we would expect for these 3 bacteria. So we know they're growing great on the supportive media. And now we can look at what we see on our selective and differential media. So let's start off looking at our selective media, the PEA agar. So this selects for gram-positive organisms, and it should prevent the growth of gram-negative organisms or really suppress it. And this is exactly what we see. So we see our Staph epidermidis, often abbreviated Staph epi. We see our Staph aureus growing on the PEA, and we see a little tiny bit of E. coli. So this is not uncommon to see a little bit of the gram-negative kind of peeking through and able to grow on this selective media, but it's really, really repressed. It's probably really hard for you guys to even see that there's a faint kind of haze there of little tiny colonies of E. coli growing on that PEA. So let's move on. So we know our gram-positives are selected for on the PEA, but let's look at a selective and differential media. So now this media, mannitol salt agar, or MSA, is going to support the growth of gram-positive salt-tolerant organisms. And that is really going to be just our Staphylococcus species. So we have Staphylococcus aureus, and we have Staphylococcus epidermidis growing on this plate. It's a little hard to see the staph epi. They're small colonies, but they're there. You can see that the E. coli does not like this media at all. There's not even a little bit of breakthrough growth. There's no growth at all of the E. coli on the MSA. What you can see, though, is that differential part. So the selective part of this is the salt. The differential part is the mannitol in that media, the sugar. Only Staph aureus can ferment it, producing acid products, whereas Staph epidermidis does not. What happens when you get the acidic products is you get yellow change of the agar. So there's a pH indicator there that goes yellow when it gets more acidic. And this is shown so nicely with the Staph aureus there, where it's turned the media surrounding those Staph aureus colonies a beautiful lemon yellow, whereas the typical shade of color for MSA is this cherry red. So definitely it's differentiating. They both grow, but it's differentiating between Staph aureus and Staph epidermidis. So that's a really nice example of selective and differential media. Let's look at our last one. I might have to open this plate because it's hard to see otherwise. So this is our EMB. So our eosin methylene blue plate actually selects for gram-negative organisms in this case. Let me just flip it over, and there you can really see that iridescent sheen on the gram-negative E. coli that are growing. It's hard to see the quadrants because it's such a, or the sections because it's such a dark media. So it's a little hard to see. I'll move it a bit closer. But there I'm really zeroing in on that section that has the E. coli. And when that E. coli produces acidic products of, in this agar, you end up with this shiny iridescent gleam to it. And it's hard to see, but the actual colonies themselves are not creamy white like you would expect for E. coli when you can actually see individual colonies. They are black. So they have black centers, and when you're really getting a lot of those acidic products of fermentation, you end up with that iridescent sheen. So that's the differential part. If we had another gram-negative organism that was not going to ferment to produce those acidic byproducts, you would see that it wouldn't have that iridescent sheen. But this media is also selective. So when we pan over to our sections where Staph aureus, so there's Staph aureus or Staph epidermidis of being streaked out, you don't see any growth. There's a little haze there for that gram-positive organism, but it's mostly probably just from the initial inoculation. It's really not growing there. So this is an example of that selective and differential. Selects for gram-negative, they're the only ones that grew on here, and then some of those gram-negative organisms, you're going to be able to differentiate them when they produce this beautiful iridescence or the black centers to the colonies. All right, and that's your little tour of known bacteria in terms of looking at them on selective and differential media.